All right, hello everybody again. Welcome back. Here is Amy Rosenfeld. Hi, Amy.
Hi, Vincent.
Everybody here, everything good this time? No echo, volume good? Is everybody happy?
Much better. I'm happier.
Well, you don't have the echo, right? But neither do they.
Yes, but therefore, I am happier.
Okay, everything seems to be good. Let's start here. See, I can put the comments up, Amy. Can you see them?
I see Jupiter crash. I like his little red spot.
My sister has to take thimerosal-free flu shot. Do any of these vaccines contain thimerosal? So only the multi-vial, the multi-dose flu vaccine has thimerosal. All the others don't. That's just as a preservative. So if you're worried, just tell them, just take the single dose vial.
I thought he meant do the coronavirus vaccines.
No, I don't believe any of them have thimerosal.
There would be no reason to.
No. They're going to be used up right away. All right, Eric. Can antibodies bind angiotensin, which one assumes has a similar structure to the virus spike, and prevent it binding to ACE2?
No. I don't think angiotensin has the similar structure. I think it has a different binding site on ACE2.
Yes, the binding site is different. That's right.
So therefore, antibodies to spike would not interfere.
Okay, here's Eric, who says it sounds great.
It's good.
Eric has been in the TWIV studio before.
Does he have a picture at the wall?
He does, but he also made me a painting of a virus, if I remember.
Yeah, we're going to have to take a picture of Michaela at the wall.
You bet. Vaccine, enhanced vaccine, antibody-dependent enhancement, a long-term possibility. You know, it's a possibility because there's a coronavirus vaccine, I think, in animals. I forgot, cats or pigs that does it. Cats.
But Perlman doesn't think that that's a possibility, and Barrick and Perlman have not seen anything like that in any of their studies or against any antibody-enhanced disease in any of the coronavirus models from SARS to MERS to COVID-19. So it's highly unlikely.
Okay.
And the cat coronavirus is slightly different.
When would we see it, Amy? How long would it take? Like within a year after vaccination, probably, right?
Yeah.
Okay. Enoch says, oh, yeah, the Lancet paper on the AstraZeneca Chadox vaccine just came out on Friday. 30% increase in low-dose priming. Why? Well, I think it's because you use a low dose, you get a lower antibody response against the adenovirus vector. And then when you come in for the boost, you get a better expression. And when you do too high doses, you get a lot of antibody to the vector. And that's known to be a problem. In fact, these 2-dose adenovirus vaccines, I think, are going to be somewhat of an issue. What do you think, Amy?
I think that the adenovirus vector vaccines will be an issue, but I'm not sure that that's the right logic.
What do you think the logic is?
So there have been some other suggestions that have to do with the way that the body responds to spike and antibodies. And I believe that Brianne on TWIV 2 weeks ago has already addressed this.
Okay. I think it's the vector, frankly, but...
Well, we'll see.
We'll see. Yep. All right, John says, now that we have effective vaccines, can we have others approved only with correlates of protection?
Can we have other vaccines approved only with correlates of protection?
Yeah, in other words, they induce antibodies. Can we license them without an efficacy
No.
test?
No.
Yeah, I think it's too soon. I think,
well,
it's only
first
2
of all,
months.
First of all, Shane Crotty would argue that it's actually the T cells that are most important in clearance. And you have had Shane several times on TWIV. So if you're just measuring antibodies, you're missing T cell responses completely. And why would the data from these vaccines or anything else be applicable to another platform? Sounds like comparing oranges, pineapples, grapefruits, and potatoes.
You mean apples and oranges?
Well, if you have 5 different platforms, you need more fruit.
You need more fruit. Okay, got it. All right, Troy, can you do a special segment that makes a coherent argument about how public research made mRNA vaccines possible? Absolutely. You know, the whole idea of making mRNA in vitro came from basic research, so that's a good idea. We'll do that.
Yeah, I did like it though. Somebody told me when they claimed that I didn't understand bioinformatics, that it was because of a bioinformatician's experiment that the mRNA vaccine was made possible.
Oh, no, that's not right.
Yeah, well, you know, what can you say?
David wants to know, how's the Chinese attenuated vaccine made? My understanding is that they are recoding the genome, right? So you change all the codons of all the proteins, of all the amino acids, so that they code for the same amino acid, but the sequence is different. And it's been shown for polio and other viruses that attenuates pathogenesis. But that's going to take a while because they have to test it.
Yes, but it doesn't prohibit recombination events from occurring to reintroduce the virulent sequences.
Right.
And coronas undergo way higher recombination rates than picornas.
I was on a call last night with a professor in Beijing, and he said they're not rushing to vaccinate because they don't have enough cases to warrant it. So they're taking it slowly.
Well, they had a stringent lockdown, and I believe that anytime that they have a case, they test the entire city, and then they do contact tracing, and then they decide whether or not they need to shut down that area, which is completely not what happens in the United States, number one. But there actually was a Lancet paper that I sent you and Daniel that talks about how Slovakia and I forget what the other country or city was that is actually doing rapid antigen testing throughout their entire population and continuously cycling through to do that so that they catch any outbreak before, which would be the smart way to go.
So Charles says that last night someone said on CNN that the molecular tests are better than antigen tests, too many false positives.
Okay.
So you don't use an antigen test to see if a person is infected. You want to do population level things, right?
Yes.
To know how who's infected at any given time. But PCR is the best, but it's too sensitive, right? And it will give you a positive, and you're not actually transmissible. So that's the problem.
It'll give you a positive. So the problem is that people don't understand what the PCR test is. And so you really can't compare. So it depends on also the primers. If you use different primers, you'll have different rates of non-specific priming. So you will, at certain cycle numbers, you will amplify cellular RNA or bacterial RNA, whatever else is in your sample, and it will be non-specific. And then when you see a fluorescent signal, it's not going to say to you, oh, I was amplifying something that wasn't really what you want. That's why when we did ours, we did it on a gel. We looked for specific base size.
So let me clarify. It says here molecular test is better. It depends what you want to do. Let's say you're sending your kids to school every day in the morning. They could take an antigen test. And if it's positive, then you're not sure it's a real positive. You stay home, you take it again, maybe later in the day, the next day. If it's still positive, then you get a PCR test. But if it's negative, they can go to school. That's the idea. But we're not having these rapid antigen tests in mass quantities anyway right now.
Despite your guarantee on TWIV, when I was there in October, that we would have a rapid antigen test by the end of the year. And my response was, what again?
Yeah, good thing I didn't bet you anything, right?
Damn, I could have made a lot of money, you know.
I don't have a lot of
Oh,
money.
and all
So
I can tell you is that headwind.
Juliet says, I've heard that exposure dose affects severity of disease. This strikes me as total poppycock. Yes, you're right. It is poppycock. There is no evidence. As far as I can see, there are no papers which say the more dose you get, the more serious the disease is. No one can quantify that. They can suppose maybe this is true. So they say, oh, healthcare workers who are, infected continuously with a lot of virus, they get very sick. But there's no proof. And I don't believe it anyway. I just think if you had more virus, it could start the infection sooner. But we don't even have evidence for that. So you're right, Juliet. It's poppycock. What do you think, Amy? Is it poppycock?
Absolutely.
So Jupiter was talking about SARS-CoV-2 vaccines with thimerosal, and as Amy said, they do not have thimerosal. Because, the thimerosal is there. If you thaw a vaccine, you keep it for a week or 2 weeks, you keep going into the vial, you could contaminate it. But that's not going to happen with these. They're going to be used up right away.
Well,
What do you think?
Not only that, the flu vaccine is not an mRNA. You wouldn't use the same preservative for an mRNA as you would use for the flu vaccine.
Right. Why is SARS-CoV-2 more transmissible than flu? What do you think, Amy?
How did we measure transmissibility?
R naught. It's an estimate, right? Statistical estimate.
Yeah, well, as you increase our immunity to it, then you reduced R naught significantly. So I'm not clear that it's actually more transmissible than flu.
I think the problem is that nobody is immune at the beginning of this outbreak. And so
Well, that's what I just said.
Is that what you said? Sorry.
Yeah, if you weren't typing and paying attention, you would have heard me.
I'm looking for a slide of R naughts. That's why.
I don't need a slide of R naught. It's just very clear that when you have a completely naive, immunological naive population, everybody gets infected. When you have people who are able to ward off and have some immunity of the infection, the R naught drops down significantly. And the flu every season is not so significantly different.
Here's a slide. Let's see if I can make this work. What do you think? Here you go. So this is R naught here on the bottom. And so 2009 flu is actually in line with SARS-CoV-2. So SARS-CoV-2 is 2 to 3. So 2009, 1.4. The 1957 and 18 pandemics, 1.8. 1980. So it's really comparable. Okay. Let's see, where were we? Frank, your podcast should be on MSM. What's MSM?
I don't watch television. I don't know.
Even if only PBS We Twiv listeners knew things months before reported on MSM TV, not enough people read Dr. Griffin's update would have scared people into wearing masks. Thank you. Appreciate that. We're just a cottage industry. Oh, this is an interesting question. What's the average amount of cells that an mRNA vaccine infects? With quotes, which is great. I know that Moderna and Pfizer have done this experiment. They've actually labeled RNA and put it into experimental animals. And, it's not billions, it's not millions, it's thousands of cells. It's not as much as you would think, but that's enough, remarkably. Oh, look at this. This is Juliet again. My sister posted a link to Mina. I guess that's Minna, New England Journal paper on Facebook, and they took it down, flagged as misinformation. Boy.
Well, don't get me started.
Here's a little clarification on there's Steen from Rutgers. ACE2 removes one amino acid from the octopeptide angiotensin 2 to yield angiotensin, so I think there's a little chance of anti-spikes cross-reacting. Thank you. Sharon's looking forward to echo-free Amy. You got her. You got her.
You'll have me for 15 more minutes. Then it's time to write a progress report.
Christina, thanks to everyone involved. I'm just a tool and die maker, but I've learned so much. Better informed tool. You know, this is an arc on TWIV. We're all just a something, right? Thanks, Christina. Any estimate on how long it will take to know whether any of the vaccines are sterilizing? Any data on that? No data. They haven't been checking. We talked about this today on TWIV. They're only looking for disease.
I don't think we're ever going to see sterilizing immunity anyway.
Do you think they're going to look for it, Amy?
Eventually. But it would be highly unusual for you to form sterilizing immunity against a respiratory disease, especially when there's no viremia. I mean, John Udall has talked about this intensely.
Mm-hmm.
And basically half your questions and half the indicators or prompts for his answers were based off of conversations where I yelled at you.
Mm. You're like Abbott and Costello, huh?
Pretty much. Can I? Which one are you? Are you Abbott or Costello?
I'm the short guy, Costello, because he's Italian. That's me. Here's one from Nick. Worried about syncytin 1 homology to spike. Would immune system select against all self if they're so restricted in tissue expression? So you're worried about the antibodies that we generate to spike having an issue with syncytin. I'm not sure it's exposed, right? That's the key. What part of syncytin is the homology? That would be the question. I just don't know. Do you, Amy?
No, not off the top of my head.
Here's a question about zinc. Can zinc enhance viral replication?
Zinc is usually an antiviral. That's why it's added to lozenges. Although it's generally thought of as an antiviral. Remember all those zinc experiments that we did?
I don't want to remember them, Amy.
Hey, I did well on that grant. I didn't miss that much.
You know, John wants to know how many mRNAs are in each nanoparticle and how many mRNAs are taken up by cells.
Well, the entire nanoparticle is taken up by the cell.
Yeah, the entire nanoparticle. And I think there can be many. I'm not sure the exact number, but you basically you make RNA and you mix it with the lipids, 5 or 6 different lipids, and they coalesce and encapsulate. So there can be many. There could be hundreds, I suppose.
For sure.
But you don't need that many to. Amy, if you introduced, say, 100 mRNAs into a cell via a nanoparticle, would they all be translated?
If they're all in, yeah. But they might not all make full-length protein. It depends on if they're intact and they have cap structures. There's very little correlation between the absolute number of RNA molecules in protein because you can translate an mRNA hundreds of thousands of times.
One way or another, response to spike will be made either by vaccination or infection. This virus will infect the majority of people in the next couple of years. I don't know if it will. If many people are vaccinated, it won't infect them, but everyone will have an immune response. You're right. I don't know how many years it'll take, but eventually most of the population will be immune. The virus will still circulate, but probably won't cause extensive disease.
Well, not in old people. It'll cause disease in children.
Yeah.
There'll be a childhood cold virus.
Susanna wants to know, would you consider the CT value useful for contact tracing? What do you think, Amy?
Absolutely.
How so?
So if you have a very high CT value, then you're not shedding any virus. You're just detecting broken RNAs.
So you don't have to contact trace those people.
I don't really need to worry about who you were in contact with. If you have a very low CT value, then you're shedding a ton of virus. Then I need to worry where you've been. But it's very hard to get CT values. It's actually very hard to get a test here at Columbia. I'm still waiting.
Oh, look at this one's for you, Amy. Hello from Albania. Is SARS-CoV-2 neurotropic? This is Amy's specialty, folks.
My specialty is neurotropism.
Yeah.
Yeah. So we discussed this on Friday on Twitter. So if you are able to infect neuronal-derived cells, or cells including astrocytes, oligodendrocytes, and neurons, you are neurotropic. So you don't have to infect the neuron directly, but you can infect all of those supporting cells, and then you would be considered neurotropic. But an epithelial cell is not considered neurotropic. It is not a neural cell. It really has to do with the three lineages of germlines, and neural cells all come from ectoderm.
So they had some evidence of limited neurotropism in that paper, right, Amy?
Yes.
So,
But Tyler's reviews it well, because you develop Guillain-Barré syndrome, you get encephalopathies and other stuff.
yeah. Can a vaccinated person that gets reinfected shed virus enough to be contagious? Possibly. We don't know, because as we said, we're just not sure if the vaccine is going to prevent reinfection and shedding. It might, because as I said, when you get polio vaccine and if you're infected with polio, you shed polio. And Amy thinks it. Sorry,
I'm not sure why getting vaccinated would prevent reinfection.
the goal is just to prevent disease, right? It's an important point. A lot of people don't seem to grasp that.
Well, it's a subtlety for the virologists. It's not a well understood difference in the lay public. I have this conversation with my family all the time.
Yeah. Someone told me today that even doctors don't get this. They think it's going to be sterilizing, but it's not.
Yes, doctors do not get this. As I said, I have this conversation with my family all the time.
Yeah.
And then I'm told I don't know anything. Then I say, well, let's just talk about shoes.
Antea, can a vaccine elicit a cytokine storm-like response?
If it's adjuvanted, for sure.
Well, it will induce cytokines. The storm part means it's pathogenic. And so if you think fever and muscle pain, fatigue is serious, then yes, it's a cytokine storm. Have you heard if the anaphylaxis observed in 2 NHS National Health Service recipients of Pfizer virus that's in the UK were allergic to vaccines or just any severe allergy? I do not know the details of that, but hopefully we can find out. I agree, it's important. Eric is excited for the prospect of mRNA vaccines against other pathogens. Yeah, I am too. Moderna, I know, has cytomegalovirus and HIV vaccines in process, mRNA vaccines. Although I don't know about HIV vaccines. There's so many have failed. Right, Amy?
40 years of failed vaccines.
ADE can't really be known by definition. Researchers say they don't think it will be an issue. That's right. I mean, for Zika, they found ADE in the lab, but it was never demonstrated in people. So you can find it in the lab, and in cells, you can find it in animals, but you have to find it in people.
How do you find it in cells?
You take a cell where the virus doesn't infect, and then you add antibody, and that allows the virus to get into that cell, which has FC receptors.
I don't know that I would say that that's ADE, but okay.
But I tell you, people published it.
Yes, I'm aware. I'm aware of exactly who published it and the detrimental effect that it had. I'm aware.
Would an attenuated platform provide a more robust immune response than just spike? I've always thought so. I was always skeptical of spike, but look, I'm wrong. You get at least short-term protection. But if I had a choice, I would like an attenuated vaccine. But we don't have a choice here.
Coming from the one who is not supportive.
Of what?
One idea that, I floated around.
Oh, Amy had an idea for an attenuated vaccine, yeah. I thought it was a good idea. Don't say that. Jennifer, how is recombination different from mutation? Amy, take that.
So mutation is generally considered a mistake that the polymerase makes while it is replicating. Recombination is where strand switching occurs and the template is the same. You have two different templates in recombination, and one template could have different code sequences compared to the other template, where a mutation is where the polymerase generally misincorporates a nucleotide. It's different.
Yeah, recombination is a combination of two different genomes that are different.
Right.
Oh, here's the lysosome. He couldn't hear you last week because of the echo. He wants you to explain the lysosome hijacking again.
So the paper was based off, the Cell paper was based off of MHV, which is a beta coronavirus studied, well characterized by Susan Weiss. And it has different accessory proteins and various things, and it's not completely analogous to SARS-CoV-2. Mm-hmm. So the paper suggests that instead of going through the secretory pathway, ER to the Golgi, trans-Golgi, and then out the general secretory, and exosome or autophagy pathways, that this goes through the lysosome. And she did one experiment with SARS-CoV-2 in E6 viro cells, which are not the right cell type. So if she wanted to make her point, she should have used Calo3s or some other respiratory cell line to make her point. But lots of things go awry in viro cells. As we know, remdesivir didn't work in viro cells, but it works when you infect Calo3 cells with SARS-CoV-2. So it's kind of a sloppy experiment and a sloppy conclusion. There should have been a lot more work done.
Microimmuno wants to know, can you take both Pfizer and Oxford? They're actually doing a study next year. An article came out today in the Times, the UK journal, the UK newspaper, saying they're going to do a trial to see if you can mix and match Pfizer and Oxford vaccines. So stay tuned. Eric, PCR is sensitive but more expensive. That's right. Antigen is useful because it's cheap. So you may have, if you have a higher false positive negative rate, you can do more often. Exactly. Yeah, which is kind of what I said, right?
Right. But it's not about the cost. It's about the fact that PCR we have optimized and the fluorescent detector is extraordinarily sensitive. And a lateral flow and protein interactions are not necessarily optimized for the tightest interaction, and they don't need to be when you're sitting in a cell and you want to do something. But in order to get them to be tighter, you can do a lot of manipulations about epitopes and various other things, but we are not there yet.
Before you go, I'll skip to this LAMP PCR question, Amy, because this is up your alley. Is LAMP better than PCR to test?
I don't know. I don't know why it would be. I mean, it's faster.
It's faster, and you can make a cheaper assay. Those New York City things on the testing centers are all LAMP tests, right? 15 minutes.
Right. But you
still need a machine, right?
You still need a machine and still some kind of amplification.
Okay.
It really all depends on the polymerase, but most importantly, it depends on all of those that you use, the primers that you use. So.
Jonathan, my wife's been in China since the pandemic. Case counts are mostly zero. When a case comes up, tracing is a national story. Wow.
That's what I said.
Yep. In the UK, we're told if we have serious allergies, we should miss the Pfizer vaccine. Is it common for serious allergies to cause problems? P.S. Thanks, Amy.
My pleasure.
Yeah, if you have serious allergies, vaccine is contraindicated. So the extent of your allergy has to be assessed, so you should ask your doctor that. Are you going to go, Amy? I don't want to kick you out, but it is.
No, I have to go. I have to write a progress report. Otherwise, we'll
Okay.
get no money. You can't
Look at
have
that.
no money and do experiments. They
I
just,
understand.
you know,
Thank you,
my money
Amy.
is dead for
Thank
the
you,
season.
Amy. Thank you, Amy.
My pleasure. My pleasure. See you later.
See you next week.
Yeah, I'll be back.
Okay, bye.
Bye.
You can you have to leave yourself. All right, very good. I could kick you out, but I don't want to do that. Okay, folks, now you're stuck with me. Susan, my pulmonologist assured me I'm safe from infection indoors if everyone is masked and distancing. Never 100% safe, Susan, because people may not be wearing masks properly. If the air is not circulated well, virus will remain indoors. So you're never 100% safe. You should be careful. I wouldn't stay indoors with a lot of people. Which vaccines are human adeno versus chimp adeno? So the Chadox one, the AstraZeneca Oxford, is a chimp adeno. The J&J is a human adeno, and there's an adeno from China, which is human adeno type 5. So, and those are the ones that are most advanced. There are others in the pipelines, but they're pretty far behind. Didn't Pfizer have people with significant allergies in their phase 3? You know, I just post. The TWIV that goes up today. There's a link to the document that lists everything. It's 97 pages long. You can almost see every patient and what they had. So check that out. You know, microbe.tv/TWIV for TWIV 691. I don't recall. I didn't get through all 97 pages. Initial viral dose was important. Yes, of course. If you put a low dose in an animal, it gets cleared right away. If you put more, it doesn't get cleared. That's the extent. But in people, that's not been done, obviously, and it's just infection, not severity. Will we be able to glean any additional info about routes of exposure from vaccine studies, or do you feel like all avenues are known? I don't know. I'm not sure what you mean. Routes of exposure. You mean the way the virus is transmitted? A vaccine is going to protect you from disease no matter what the route is. So I'm not sure that's going to tell you anything. But I may be misunderstanding. You can re-ask it later. Yes, of course. Asymptomatic transmission plays a big role in spread. Of course. Although it's probably hard to get data about this, can asymptomatic people who have a diluted immune response have an immune response if they're infected again? We have so little data on reinfection. They could, yes. So it could be like a boost the second time, right? I mean, but one of the ideas about asymptomatic infections is that these individuals actually have a really good innate response that tamps the reproduction down so it's not symptomatic. We're still trying to sort that out. Yes, of course, R naught is independent of pre-existing immunity, but what you do to calculate it, you look in. If you want to know the absolute R naught, you look early in an outbreak before any mitigations, before any population immunity. You then can calculate it there, because as soon as you start masking and sheltering in place and so forth, then you affect the R naught. Pfizer vaccine is a multi-dose vial. Yes, that's preservative-free. Exactly. It's got to be shipped frozen. Okay, thank you. MSM is mainstream media. Yeah. You know, MSSM is Mount Sinai School of Medicine. Thank you, folks. I know we're a little bit behind. What the weather is like, I don't know. I'm in the basement. It did snow today here in New York area, although it didn't stick. So here's a question a lot of people bring up. What's the difference between the rushed vaccine approval process and the typical? So typically they take a long time because we don't have a pandemic that's killing so many people, right? So there's an impetus here to try and do our best in a year, which I find is amazing, but they were able to coordinate it because these clinical trials are not easy to put together and execute. It's just mind-boggling to me. So, but they've done phase 1, phase 2, phase 3. They've collected safety data all along. They've looked at efficacy. They've looked at immune responses. And, for the Pfizer and Moderna, it looks good to me. The AstraZeneca, I can't tell because they haven't been straight with us with the data. But hopefully others will be straight coming forward. Tomorrow the Pfizer vaccine is going to be reviewed by the FDA, so we'll get some more information from that. And so normally it would take years. You would let the phase 1 go for 6 months, and then wait and analyze the data, take your time, and then the next year do the phase 2, not overlapping. Right? So it's all accelerated. What's your thoughts on research on vaccines versus antivirals? I know a lab that specializes in antivirals, but I can't imagine any of the drugs ever receiving. So the vaccines are ahead, right? And what they did with antivirals is they checked some FDA-approved ones or things that had been in people already. So several of the HIV antivirals were checked, remdesivir and others, with the idea of getting them out quickly. But they don't work. They didn't work in very sick people. You have to give them early, and it's really hard to give to people very early in infection because, if you're in a hospital and you can't breathe, then it's too late to give you an antiviral. So antivirals are tough for this virus. For flu, as soon as you have a positive flu test, you can go get Tamiflu within a day or two after symptom onset, and then it works. And you'd have to do the same with this. And we simply don't have any antivirals that fit that. Now, they're in development. People are working on them. And I suspect they may be ready to test next year. But they're certainly behind. Costello was Irish. Okay. The name kind of sounds Italian, right? My family met him once, so all right, I could be wrong. Sorry. It's not my forte. Regarding innate immunity, is it common to have so many viral proteins to interfere at so many different levels? Well, it varies on the virus. Some viruses have 1, some have 2, some have more. And these genomes are big. They can encode a lot of proteins. So I don't know. There are no rules, right? It's just what happens. What's sterilizing immunity? Sterilizing immunity is the virus enters you and it doesn't reproduce. It's completely blocked early on by the immune system, right? Maybe it goes in a few cells and then the antibodies or the T cells get rid of it. As opposed to what most vaccines do, the virus gets in, it reproduces for a few days, you can shed it, and then your immune response kicks in and gets rid of it. So obviously, if the virus can reproduce, it has the potential to spread, even if you're immune and not getting sick. If syncytial cross-reactive immunity were an issue, we probably would have seen it in common cold coronas. Maybe. I don't know. I'm learning that every corona is different. I never expected this one to do so many things, frankly. Oh, yes. Indirect acting antivirals. So antivirals that target the host. Well, there are some good ones, right? Let's see. There's the CCR5 antiviral for HIV. So they can work, but of course CCR5, 16% of the population lacks it and they're fine. So it's obviously not essential. It's redundant. So you have to pick a target that's like that. And it's hard to do. It's really hard to do. And companies are wary. They're afraid that they will take the antivirals a long way, spend a lot of money on them, and then they get toxicity in people and they have to can it. So understandably, they'd rather have a direct acting antiviral. That's a good definition. Sterilizing means you'll never ever catch it, not even to spread it. New study out yesterday shows biomarker for blood vessel damage in all children infected, including asymptomatics. Interesting. Very interesting. I'll have to look at that. What journal? Put a link in if you can. Netflix Epidemic series talked about making universal antibody for any antigen. I don't think you could do that. There isn't an epitope shared among all, say, viruses. You could make a universal flu antibody or even corona or even HIV, but they're going to be different. Okay, Elise wants to know if is this going to be a problem with keeping the Pfizer vaccine cold? So this is a good question. Pfizer is shipping these in suitcases full of dry ice, right? Which will last for 5 days. Then you have to fill them up again if you don't put it in a minus 70 Celsius freezer. Not everybody has those freezers. Not even in the U.S. And in hot parts of the world, they don't have them. And many parts of the world don't have dry ice, right? So I think it's going to be a problem. And the Moderna can last a few days at least at refrigeration temperatures. So this could be an issue. I agree. They're saying it's not a problem, but for these reasons, I think it could be an issue. Okay, Bradford says, didn't AstraZeneca test for sterilizing immunity? So I've heard that, and I haven't been able to find the data. They haven't really released those data. I heard that they did, but I don't know how. And certainly Pfizer and Moderna did not. I checked very carefully, and they are not doing it. So Carl thinks that measles vaccine is sterilizing. I asked this question of Sally Permar a couple of weeks ago on Immune, and she said, no, it's not sterilizing, because that's what I had heard too. So probably not. Dinah says, could new SARS variants associated with farm mink reduce effectiveness of vaccines? Yeah, impossible. It's possible. So there was an isolate from Denmark with 4 or 5 amino acid changes that was not neutralized as well by human convalescent sera. Now, it's not clear that the difference would impact vaccine efficacy, right? Because those are in vitro experiments in cells in the laboratory. I don't know how you can extrapolate them to people, but in theory it could. And so I think it's really important not to have big reservoirs of the virus like mink. I think you need to make sure they don't get infected. It can be done. You can make sure the workers are checked until we have a vaccine. You could even make a vaccine for mink or whatever other animal that you raise and you want to protect. It's not so hard to do. So I think I'm concerned about it, but right now I think we're okay. So the masks reduce the viral load. How can it be poppycock to say the amount of exposure doesn't relate to less sickness? You reduce the amount of virus and you don't get infected because you're now dropping it below the threshold where the immune response takes care of it readily. It's not about, maybe we're just confusing our nomenclature. It's not about how sick you get, whether you have an upper tract disease or severe life-threatening COVID. I'm just saying whether you get infected or not depends on the dose. I agree with that. How does the dendritic cell differentiate between the spike and cellular proteins degraded by the proteasome? Well, it doesn't. It just puts them all up on the surface, right? Whatever it gets, even cell proteins, it puts up on the surface. In fact, they routinely sample cell proteins, and then the ones that are foreign are recognized by the T cells. So the dendritic cell is not discriminating, but it does take up the liposomes, the RNA nanoparticles, and so those proteins are going to be made inside those cells. Eric's lab is exploring S2, this S2 subunit of spike. So S2 appears to have some broadly conserved regions, right? And it's been suggested that actually kids infected with common cold coronas have antibodies that neutralize SARS-CoV-2 by virtue of S2 specific epitopes. It's a possibility. We'll see. I think people are clearly, in your lab and others, are going to explore it. In the early days, there were some papers suggesting GI symptoms. Yes, for sure. There's no doubt. Daniel Griffin mentioned many times a lot of these patients would have GI symptoms before respiratory symptoms. And so the idea is that the virus might be reproducing in the epithelium of the gut. Now, very few people have isolated infectious virus, though, from feces. So I'm not sure we really understand what's going on there. It is early. Still, it's less than a year, right? Will the mRNA continually create spike proteins? So Pfizer says the mRNA lasts about 2 days. It's got a turnover, right? Like any mRNA, it doesn't last forever. It's degraded by cellular RNA degradation systems. And so this will last, they say, 2 days, and then it's gone. It's made its proteins, and then you're making your immune response. Okay, so these are all about the amount of virus and getting infected. Do mRNA vaccines require targeting antigen-presenting cells? No, they don't actually. As you say, the muscle cells can present them too, sure. So when you get it injected in your deltoid muscle, most of it drains to the local lymph nodes, which are in the armpit, right? And they're taken up by APCs. But some of it can get taken up by muscle cells, which would then present them. The muscle cells might die, and then the pieces would be picked up by antigen-presenting cells, or they could be taken up by antigen-presenting cells that are patrolling the muscle. So yes, they don't have to go into the APC. Oh, this is an interesting question. My understanding is that Moderna has been researching mRNA vaccines for the last decade. When did Pfizer start doing this, or did they jump onto Moderna's research? I've wondered the same thing, okay, that Pfizer just jumped on. I don't know the answer, because it's kind of suspicious. Moderna's been doing this for a long time, for over 5 years, and they've had other vaccine candidates being developed. And so all of a sudden this year we have a pandemic and Pfizer's making mRNA vaccines. Now, theirs is different from Moderna's. The composition of the nanoparticle, the lipids that they use are different, and that's why Pfizer's has to be kept at minus 70 Celsius, and Moderna's does not. So they're obviously different. Now, earlier this year, Moderna lost a patent claim. They said that their idea of making mRNA vaccines was theirs, but Pfizer won a decision that it isn't. So, Pfizer didn't do it first for sure. Moderna did. And I mean, I think in a pandemic we need to have everyone making it, but, it's not great. I agree. 3,200 deaths in the last 24 hours. Yep, 9/11 each day. And this is really the post-Thanksgiving holiday bump, right? And then we're going to have another one, Hanukkah now, and then Christmas and New Year's. It's really bad. Vaccine-associated disease, right? So there's antibody-dependent enhancement and vaccine-associated disease. They're both plausible. Barney Graham wrote a nice review article about this in Science a few months ago. He said, yes, they're always a concern, and that's why we have to test and look out for them. And so we have had 2 months of testing, which is not a lot, and it's not huge numbers, so we have to keep looking for this. But it's a trade-off, right? It's a cost benefit. You've got 3,200 deaths a day, and really we need a vaccine because obviously masks and distancing isn't stopping that. Now, I don't know if I can answer this. In diabetes, is it higher blood glucose or higher insulin levels that presents the greater danger? I don't know. It's just having diabetes, right, is a comorbidity. Why so? I don't know, and I'm not sure anyone knows. Is there a concern for newly vaccinated healthcare workers to shed to their patients or coworkers or family members? So we talked about this today on TWIV. If you get the vaccine, it doesn't mean you can rip off your mask and go about life as usual because you might still be able to be infected. So healthcare workers, of course, are at high risk for being reinfected. So until we know more, we do need to be very careful. And I know people don't want to hear that, but until we know that vaccination cuts down transmission, you have to be careful. A friend of mine said he heard the mRNA vaccines may cause a massively worse symptom for the people who it is not effective for. Absolutely not. Everyone who's received this so far. So the numbers are out there. You can look at that Pfizer report. The biggest side effects are fever, fatigue, and pain at the injection site. And, a third of the placebo recipients also reported fatigue because people are tired, right? So I don't see any such massively worse symptoms. No, this is a kind of rumor that isn't true and just trying to discourage people from getting it. Oh, yes, Brian, I saw Florian Kramer's phase 1 trial on this universal flu vaccine. I think it looks good. It induced good neutralizing antibodies. I'm thinking of getting him on TWIV to talk about it. I think that's really important. I'm sorry. I like Paul Offit, but I don't see any dose severity data. I'll look at it. I'll go look. But I'm not going to go see him on this podcast. I'll email him because I know him. Could you explain how human behavior in different seasons affects contagion? In my country, cases are reducing. We are in spring to enter summer. So here's the thing. There are two major impacts of season. First is that people in a cold season, they go inside and they congregate, and it's easier to pass an infection when you're inside. For some viruses, though, for the respiratory viruses typically, but for polio and other similar viruses, they actually spread better in the summer when kids are outside playing with each other. So that doesn't hold for all viruses. But the other one is, in the winter, cold temperatures maintain virus infectivity longer in droplets in the air. And the low humidity allows the droplets to travel farther. So when you talk, you expel these droplets of moisture which contain virus. And when it's humid, they absorb moisture and they fall to the ground quickly. In the winter, less humidity, the droplets stay suspended, better transmission. Okay, when it's dry. And it's even dry indoors. You turn the heat on and you dry out the air. So those are the two factors, the weather effects and the people going indoors. If meat is cultured from animal cells, can a latent or dormant virus? So you're going to take cells and you can make meat out of it to eat, in other words. Yes, it depends what kind of meat. Every animal has its own kind of integrated retrovirus, like pigs have hundreds of different integrated retroviruses. And, we use pig valves for transplanting. And so people are worried about that because you have to immunosuppress the person to put the tissue in. So people are trying to make pigs free of all these endogenous retroviruses. George Church's lab is doing that. A single Pfizer dose could have been enough. So they saw some efficacy with a single dose. That's right. But it's not as good as with two. So this is a double-edged sword because some people are going to say, I'm not going back. I got one. I'm okay. And I think it's better to have two. Thank you. I really like doing all the pods. I like answering your questions. And Amy is wonderful. Yes, she's very smart. All right, Cass says, if the viral-loaded infection has no impact, why are dose rates at the high end during trials show that severity are high? Well, the two trials we know the most about are mRNA vaccines, not even infectious virus, right? So the side effects are due to cytokine inflammation as a consequence of the amount of immunogen. Hopefully we'll have intranasal vaccines. Yeah, that would be great. What I think we're going to have in 5 years are skin patches, right, where they take a little plastic square which has tiny needles in it. It's impregnated vaccine. You put it on your skin with a Band-Aid, and it injects the vaccine into the dermis. They work really well for influenza. We're going to see all those going to get rid of needles. It's going to be really good. Yeah, lamp machines are quite small, and you can. It's the Abbott one that came out first. So yes, they're smaller and cheaper than regular PCR machines, and they're faster. Can we please review this hybrid synthetic mRNA technology? So basically, they sequence the genome of SARS-CoV-2 back in January, and then you say, okay, here's the spike gene. we want to make a vaccine. So they simply copy the sequence and they synthesize it and put it into a plasmid to make mRNA in vitro. The only thing they did was to change 2 amino acids so that the spike would not be cleaved in the cell. It would stay in the prefusion form, which is where all the epitopes are. So there's no, I don't know, what software coding. All you do is print it out and send it to a company to make it. But I mean, there are many, many more sophisticated things you can do with computational biology, obviously, and sequences. But for the mRNA, you really have to do very little. Should we be wearing N95s? I don't believe so. I think a well-worn, proper surgical mask or equivalent is fine. And I'm an N of 1, but I've been wearing them the whole time. And I go to Columbia 2 or 3 times a week. I don't take mass transit, obviously. I don't encounter a lot of people, but I go in elevators and so forth. And so far, I haven't been infected. Yes, we talked about that already. Immune podcasts, you bet. I'd love to do more. The hosts, they have their time considerations. I long ago said I want a podcast. That's where I can make an impact, and I'm lucky that I have Amy running my lab. Otherwise, I couldn't have both. My dad's always banging on about Novavax. By the way, there are a lot of questions about dose and severity. Okay, we'll address it in an upcoming TWIV, I promise. I don't mean to blow it off, okay? We will look into it in detail. Novavax is making a protein-based vaccine. And it's different because it has chemicals added to it, lipids that aggregate the protein. So it's more immunogenic than just the protein on its own. And so they're slightly behind the other companies, but they're moving along as well. And that may be one of the others that's used in the U.S. So we're going to use Pfizer, Moderna, AstraZeneca, J&J, and possibly Novavax. What kind of test tells you if you can hug your family member when they get home from working in a business with big COVID clusters? All right, so any test is only good the time you take it, right? So if you take a test today and then 2 days later it's negative, that doesn't mean you weren't infected in the intervening 2 days. And that's why if we had a test that we could each take every day, then that would be the key to being able to hug people who were working into those conditions, right? So I'm afraid there isn't one right now because we don't have these rapid, cheap antigen tests. Novavax has awesome antibody but mediocre T-cell response. Yes, this is a good point, Kaz. The Russian vaccine has 2 different adenovirus strains. The first is different from the second, so that eliminates immune responses to the vector being an issue. I think that's perfect because I think if you use the same vector for prime and boost, you're going to have a problem. This is not a strat, actually. I couldn't afford a strat. This is a Starcaster, which is about $200 I bought 20 years ago. I was playing guitar back then, but I play viruses now. I'm sorry. I don't know what the Biden administration is planning. They're planning to they have a nice committee. They're keeping on Dr. Fauci. They're planning to help distribute the vaccine. I think the most important thing that has to be done now, we have federal investment has made a number of vaccines move forward, but the distribution is up to the states. The feds aren't helping at all, and I think that's where Biden could step in. We have a decentralized healthcare system in this country. Every state's on its own, basically. Some are really good and some are not. So distributing the vaccine is going to be a real problem for some states, and I think that's where he could help, in my opinion. This is not science, but it's what has to be done. even if those who vaccinate still shed, how does that affect the number of people that need to be vaccinated? It doesn't. It's still the number of people that have to be vaccinated to reach herd immunity is still calculated based on the R naught. So if it's 2 to 3, let's say it's 3 for SARS-CoV-2, that's 70% have to be immune to get herd immunity. So that's doable. German companies develop a rapid PCR that takes 39 minutes. Well, that may be a LAMP test. It may not be PCR, right? It may be nucleic acid based, but a different technology, and that's why it's faster. Standard RT-PCR, if you're going to do 40 cycles, will take you 8 hours at least. You're welcome. Virology course you can find on YouTube, youtube.com/ProfVRR. And this January, we start a new semester, new course. And funny, the last course, 2000 version in January, we followed the development of this pandemic, and now in the next course we'll follow vaccination and what it does. It should be really interesting. You want to see the R naught slide? I showed it. Do you want to see it again? Here. This is published in the New York Times. There's the link. And it's in my virology course last year. I think it's in the introductory pathogenesis lecture, but case fatality ratio versus R naught there on the x-axis. All right, what else do we have here? If an mRNA vaccine has never been approved, why do people think a rushed would be safe? I think that you are completely justified to ask that question. And I had the same issue in January. But as I said, they've gone through each of the 3 phases, and they have done the right number of people. They have done the right number of time. They've just cut out the in-between time, and they've done a little overlapping. And it all looks good. The numbers are good. They look safe as much as you can say within a year, which is not to say that there won't be issues when you put it in 10 million people. I completely admit that. And you have to decide whether you want to take it or not based on that. I totally agree. It's a risk-benefit ratio. If you're not in a high-risk occupation where you contact a lot of people, you might not want to do it, and I understand that. But I'm going to do it. I can keep myself here in the basement and podcast all the time, but I would like to start traveling again next fall maybe, so I'm going to take the vaccine. But I totally understand. I'm just saying I don't see anything wrong with it, and I have been looking at this kind of data for many years. Are there nasal coronavirus vaccines being developed? I believe there are. There's one that's an enteric vaccine. It's an adenovirus, which it's in a capsule that passes through your stomach and comes apart in the intestine, and then the virus multiplies in the mucosa. And if you make immunity at one mucosal site, you tend to make immunity at others. So that should give you respiratory mucosal immunity. It's very interesting. I don't know about a nasal vaccine. I know that people have peptides they want to spray into the nose that would prevent virus from getting in there. I don't want to keep you all here forever. We can just wind up with a couple questions here. Oh, this is a good one. Given that we'll have more vaccines in the coming years, do you think the time required to approve vaccines will change? No, I think this is a very unusual time, right? We have an urgent situation where we can't save people unless we have a vaccine, and many people are dying. So that's the rushing. So I think we're going to go back to the old method where, you can mitigate, you can do other things so that you can limit infection. I feel like people have lost their sense of logic and free thinking. I'm sorry. I think I'm thinking freely. I view all reasonable arguments. But goodbye. U.S. surpasses Argentina in deaths per million. Yep, it's very bad. Oxford and Lancet. Yes, they came out Friday. Now, but the half dose versus the full dose, it's not clear what was going on there, and I don't think they've told us what's going on there. All right, let's see. 3 more. Pfizer handling protocol includes all sorts of detail. Yes, it does. It's great, but if you don't have dry ice, you're screwed, and if you don't have a -70, you're screwed, and not everyone has both. Absolutely not. Does the vaccine give you Bell's palsy? No. I know of no example of that happening as a cause of the vaccine. Ivermectin, doxycycline, and zinc. We just talked about it on TWIV today, so check it out, okay? Our thoughts are there. That's right. Pfizer did buy the mRNA technology from whatever it is. Yes, BioNTech. Yeah, I'm confident it's not theirs. They're just assisting. That's the story. You're absolutely right. And Pfizer is a big, multinational company, so they can do that, whereas Moderna is not, and they have to partner with people. My wife got COVID symptoms today. Well, you have to get her tested to make sure it's COVID because you can get other things at this time of year. So basically, if you get swollen glands under the arm after the initial vaccination, you know it's working. Yeah. Yes. Any idea why the U.S. didn't attempt to bring any inactivated vaccines to market? I don't know. I'm not party to those, but I would have tried because it's really easy to do. And, we have a lot of good inactivated vaccines. I don't know why. A recommendation regarding indoor humidity. Keep the humidity as high as you can make it because that'll make the droplets big and drop to the floor and keep them from spreading long distances. So it's hard to do in the winter because heating takes the humidity out, right? Ivermectin we reviewed today on TWIV. It doesn't do any good if you give it to people in the hospital, and that's what they've tried it in. And plus, the doses you need are not approved in the U.S. So these are all issues. All right. According to the internet, the movie database, Costello's father was born in Calabria. His mother is Italian, French, and Irish. There you go. One more. And I'll be back next week. I'm very concerned about long haulers. Yeah, 1/3 of people get long-term COVID. And there are serious studies looking into this and seeing how to treat these individuals. Absolutely. There's so many of them that it's being done. And, I don't know if you know the disease chronic fatigue syndrome, MECFS. I mean, those people for years have been suffering in a similar way. And they feel that they've been neglected. And now COVID, long-term COVID comes and they get a lot of attention. So it's kind of tough. TWIV is This Week in Virology. You can search for it. You can find it. I do that twice a week, 3 times a week. I'm on YouTube as well. Just Google Racaniello. It rhymes with black and yellow. Hey, everybody. Thank you so much for joining me. I will see you a week from tonight, 8 p.m. Eastern. Come back, and Amy and I will attempt to answer your questions. Thanks a lot.